Mangrove forests are important ecosystems that are found in tropical and subtropical regions, mainly between 30 degrees north and 30 degrees south latitude. They can withstand high temperatures, high salinity, extreme tides, and poor soils. It is well known that mangroves are among the most productive ecosystems in the world and are extremely beneficial. Not only do these forests serve as nurseries for many tropical fish, sharks, and other sea life, but they help stabilize coastlines during tsunamis and hurricanes. Mangroves also sequester carbon, are an important food source to animals, and are even sources of medicine. The reviewed study looks at the respiration rates in the embryonic stage of 6 different East African mangrove sesamoid crab species. These crabs lie within phylum Arthropoda, subphylum Crustacea, class Malacostraca, order Decapoda, and family Sesamoidae, which include all terrestrial crabs. The study was carried out via field sampling and laboratory analysis in Gazi Bay, which is 40 kilometers south of Mombasa, Kenya. All 6 species are phylogenetically related, showing very similar reproductive traits, clutch size, and developmental time, but inhabit 3 different ecological niches. The crabs range in size from 1.5 to 3 centimeters and are common to mangroves in Africa. They are all omnivorous and can eat insects, annelids, mollusks, and algae that is filtered through their sieve-like mouthparts, but they mainly feed on fallen leaves. Here is a video showing crabs eating leaves.
Of the 6 study species, Caromanes ormani and Caromanes eulemene represented the most terrestrialized subjects. Their habitat includes shoreline covered by high tide 6 to 8 days a month, and salt-tolerant grasslands the remaining days. Parasesarma leptoma and Celatanium elongatum were representatives of the arboreal living crabs, their habitats ranging from crevices formed by aerial roots of mangroves to mangrove canopies. The last 2 species, Parasesama gautamum and Parasesama samawati, are found in the littoral zone and are active in mud during low tides. Female crabs carrying eggs from each species were reared for a week in plastic tanks containing a layer of moist mud on one side and aerated seawater on the other side. Aerial measurements of oxygen uptake in the embryos was performed by putting the eggs into gas-tight containers filled with oxygen-saturated air. To measure aquatic oxygen uptake, eggs were placed in 2 different containers. First, the same gas-tight containers were filled with air-saturated seawater, and the second method used 8 micro-respiration glass chambers filled with seawater. Oxygen saturation was recorded using a needle-type fiber optic sensor placed next to the eggs. For each test, 100 to 200 eggs from each species were removed from the female and placed in separate containers. Control containers with no eggs were also tested. After the trials, all the eggs were then fixed in formaldehyde solution, scanned and analyzed with software that assessed changes in volume during embryo development.
This table illustrates embryonic respiration of the 6 sesamoid species. Shown is embryonic oxygen uptake standardized by the mean volume of the eggs. The Roman numerals at the base of each bar represent the embryonic stage each group of eggs falls into. Group A represents respiration in air, while Group B represents respiration in water. Group Z represents air versus water respiration ratio recorded in embryonic stage 5. As you can see, 3 species, one from each level of terrestrialization, showed significantly higher rates of oxygen uptake in water than when they were placed in air. This form of adaptation, in which the embryos revert to a more ancestral historic form, is called adaptive recovery. Simoni et al. Found lower embryonic oxygen uptake in air with respect to water for all tested semerids. All species showed a similar increase in air respiration during embryonic development. In regards to oxygen uptake in water, the species saw differences between each other along the overall development, but all species did display significant increase in oxygen uptake through development at least one of the 5 embryonic stages. All embryos of the 6 species displayed bimodal respiration, the ability to breathe in both air and water. This confirms the embryos of these mangrove sesamoids are all adapted to the semi-terrestrial and arboreal lifestyle adopted by adults through evolutionary processes. The results did not prove these species to be amphibious, but they did demonstrate an adaptive evolutionary trend towards terrestrialization in the embryos.
This study shows that embryos of the studied crabs are a further step towards terrestrialization, although they are not fully amphibious. The development of embryonic respiration in this species is evolving to accommodate their movement away from aquatic habitats. Because of this, water is no longer a major limiting factor for female crabs bearing eggs, demonstrating an adaptive recovery in certain species of sessamid crabs. Changing climates and environments place a strain on species who cannot adapt to these changes. The evolution of these mangrove crabs to better adapt to terrestrialization is an indicator of how they may be able to survive in future changing environments.